Hello, my name is Jimmy Lin. I'm the founder and president of Rare Genomics Institute. Today we're going to be talking in a session called Implementing Clinical Research, Whole Exome and Genome for Diagnosis of Rare Diseases in a Global Perspective. What we are is we are a nonprofit organization that helps patients with rare diseases to be able to access cures. What we have done as a first step has been doing genome sequencing, and we're now sort of doing processes in order to um, start to do model organisms and to look for drugs and to do the subsequent functional analysis that is necessary afterwards. So I'm going to talk a little bit about what we've been able to learn through our sites, which we have mostly in the US, but we're also in countries such as Singapore, Malaysia, Australia, Israel, and the experiences from using whole exome and whole genome to be able to help these patients. A little bit of my background. I did my MD PhD at Johns Hopkins, where I trained, and with that background, what my work has been was we were the first to solve the cancer, to determine the cancer exome. So we initially did it for breast and colorectal cancers. And then eventually we were able to do similar type of studies with pancreatic cancer and glioblastoma. All this work was mostly sort of at that cusp of next generation, but this was really an ability to start even looking at, what was possible through exome sequencing. And eventually we were able to actually look at, medulloblastoma and also susceptibility, and this was, a lot of the background for which sort of I bring to this. But then my life was dramatically changed. After I finished my PhD doing all this work in cancer genomics, I met a family. This is not their picture, but it was a small little Indian family, an Indian little boy that was sick. They could not figure out what was wrong, and they traveled all the way from Buffalo all the way down from upstate New York to Hopkins to be able to find a diagnosis, but nobody could help them. And at that time, those were the first studies were out of University of Washington, where exome sequencing was starting to be used to determine causes of diseases. And we think, what if we took all of the experiences that we had in cancer and start using it for exome sequencing for rare diseases? So that was been really the start of sort of our creation of our nonprofit to be able to implement and help rare disease patients access exome sequencing, access different centers to be able to do that, have the analysis done. But also now we're definitely sort of expanding as exome sequencing is now more and more available clinically, and we covered by insurance. We now see the need sort of shifting downstream to be able to figure out what those genes do and how to find potential therapies and drugs, or how to study the function of those genes to be able to help. So for those who don't know, this is sort of a quick plot, generalized plot of diseases in general. There are some diseases that have very high prevalence, but there's a long tail of diseases that have very low prevalence. In fact, there's over 7,000 different diseases in this long tail, often considered labeled as rare disease. In the US, it's less than 200,000 patients affected. But it's very hard to be able to solve and figure out ways to help all 7,000 diseases. And it affects over 300 million patients worldwide. So this is actually a pretty large disease burden. And to be able to start sort of making an impact is a sort of a difficult problem. So because it's so sort of diverse and it's split up into so many different diseases, right now, the vast majority of rare diseases don't have a research program specifically to help them. Many of them, vast majority, don't have an advocacy group, foundation support. So most of the families are sort of on their own trying to figure out what's wrong. And now even with the proliferation of exome sequencing and clinical exome sequencing, most of these families, even if they're able to get a diagnosis, really struggle to be able to help their child that's in need. So thus, there's less than 5% of these diseases that have any treatment or therapy. And this then is a pretty serious problem, thinking that 95% of these 7,000 diseases, so 300 million people, don't have a mechanism to have any type of therapy. So that obviously is a great burden, not only to think of it as a population level, but also that every single one of these kids have faces and have names and they have families, and that's why it sort of is very, very important to figure out ways to be able to help. So this is why we created our institute, completely volunteer run by people all over the US and the world, to be able to figure out ways to be able to help these patients. So I'm going to walk through one example of a patient, and then we'll also then talk about more in general then in terms of lessons we've been able to learn from that. So let's go walk through one example. This is Robert, one of our first patients. He was born healthy, but then around age 1, he all of a sudden sort of lost control of his muscles when he was unable to feed, and had a dramatic clinical decline. So they started rushing to the hospital and started testing and trying to figure out what was wrong. And 14 years later, over millions of dollars later, Robert still really didn't have answers in terms of figuring out what was wrong with him, and still no group that belongs to help him, and they go, try and place to place, try to figure out what's wrong with Robert. But the very fact is, the system is very hard to help Robert, and this is sort of 2 years ago when there was very little access to this clinical sequencing. We started to decide to figure out how do we do this then. So the first thing that we do is provide sort of the technology, and of course we're in this talking about exome and genome sequencing. We all sort of know that for diseases like that, using whole genome or whole exome sequencing is potentially very effective. There's a picture inside of a scanner to be able to map not only sort of Robert's genome, but his parents' genome, to be able to find the gene of interest. Then we found people who are able to do this research. So like I mentioned before, we're at many centers in the US and also in the world, including Harvard and Yale and Hopkins. And we approach researchers who are willing to help patients like these, and able to provide their existing pipelines and networks to be able to help. And then lastly, although there are some funding for exome sequencing, a lot of our patients don't fit into the traditional funds that are existing. So we want to make it sort of, as we are expanding, so that all the patients are able to access it even without funding. So we have an online platform, which is a crowdfunding platform, where patients themselves are able to raise the funds that are needed for the different research projects, starting now, which is mostly now sort of based on exome sequencing. So for Robert, I think this is sort of standard prices around exome sequencing, sort of between $7,000 to $10,000, and they were able to raise it from friends and family, and this, they did not have to sort of fit into an existing funding mechanism to be able to help. So what we have then is a team of scientists and researchers at different universities, along with their community, providing the funding for this to be able to help patients like Robert. So today we actually are excited to report that we are able to sort of find out the gene of interest is PR. It's a PRKR16 gene, and Robert actually, because of that, the therapy is now starting to be able to use assistive devices due to the diagnosis. What was important is for Robert to be diagnosed in terms of knowing that his effect is a muscular effect and not necessarily cognitive. So Robert, for the first time, is now communicating with his family, with this sort of diagnosis, and be able to move on in terms of what the family is able to get. And today, of course, there hasn't been a dramatic cure yet for Robert, but the next step now, we're actively studying his gene. There's a team of scientists in Canada, as well as in Washington University, St. Louis, that are now looking at the gene that's sort of affecting him, and we are actually in the middle of starting to do some functional studies. And The goal is to not only do the sequencing, is have the full pipeline to be able to help and be able to fund every step of the way. We know as genome sequencing becomes more and more prevalent, more and more people will have sort of gene sequence, but then the functional annotation is still not there yet. So there is going to be a lot of work to be able to figure out what these genes do. We've had amazing sort of partnerships from a lot of different companies who've been able to support, providing whether it's funding or reagents or model organisms to help, as well as our story actually, our model of sort of bottom-up research has actually been able to garner a lot of press attention, so that a lot of patients sort of come to us as we sort of figure out ways to help these patients with rare diseases. So far, I think, like I mentioned, we're mostly right now in the U.S., but we have sites in Spain and Israel, and we're starting to do work also sort of in India and Australia, and then really just sort of provide the infrastructure if there's a need to be able to either raise the funding or help them with interpretation and everything that's sort of needed for these projects. So that's just sort of a quick intro as I come into the area where what is it that we sort of learn from all these different sites and all these different projects in terms of the implementation of exome sequencing. Firstly, there's discussion of sort of participant selection. So if you see this graph here on the x-axis, it goes from group decision to individual decision. On the y-axis is clinical to research. So the x-axis is how is it that someone is determined whether they're a good candidate for sequencing or not. So there are some centers that have sort of large groups that decide, and well as as we more and more clinical, you see where the start is. You want really one clinician to be able to sort of make the decision to think it's an appropriate test. On the y-axis here, it goes from research to clinical, as everything sort of startups in the research criteria, and eventually you implement it in terms of clinical context. And there's the two developments here, whether you develop it starting from a group decision clinical testing, or you go from group doing research and make it individual testing, then you need sort of criteria development. So ultimately, what are sort of the criteria that fits, especially you want to test the criteria? There are three specific sort of criteria as we implement, and this is can be generalized through a lot of things. Very important for patient criteria, right? You need to, not only the phenotyping is going to be very, very important in terms of looking not only through history of illness, like a previous testing, especially whether they've done microarray testing, and whether their phenotype is something that fits sort of existing genetic type. Family characteristics is also going to be very important, whether the pedigree is available, the mode of inheritance that you sort of suspect, as well as sort of population characteristics, whether they match similar families or known syndromes. So these criteria makes it in order to be able to determine, and having these criteria then allows us to be use it as a clinical testing tool, and no longer requires sort of these groups to be able to do that. Then in terms of depth of coverage, how deep do you want to sort of cover as you sequence? And here on X criteria, sort of look at single gene traditionally all the way to whole genome, and then the coverage is on the y-axis, going from sort of roughly around sort of 10 to 1,000. And traditional testing obviously has done very deep coverage with Sanger sequencing and genetics panels as you move more. And with cancer, you want to have very, very high sequencing. So even if you use next gen, having these high coverage is going to be important. But as you sort of move towards sort of genetics, as you sort of see sort of the green dot, then sort of the coverage is, I think for most of our sites, the coverage is anything sort of between 60 on the lower side to sort of 90x in terms of coverage from genetics. Whereas for cancer, on the exome side, we can, if it's a pure sample, we can do whole exome, but really you want it to sort of much higher because of heterogeneity in cancer. But for genetics perspective, the coverage really doesn't have in terms of depth could be lower, and that allows for you to be able to put out sort of a lot of these samples on even one run especially when you're doing sort of trios or larger families to be able to do that. So how about in terms of sample prep? What are the things that the sort of different sites do? And here you see, sort of the blocks here. There are two sort of specific ways, whether you want to do amplicon sort of methods or capture methods, and then whether you want to do exome or panel. Because due to the sort of the nature of these sort of sequencing diagnostic odysseys, more methods have sort of been thinking more in terms of sort of using a larger exome would be able to look at all unknown diseases. However, if there's sort of specific panels, for example, cardiomyopathy or hearing loss or related kind of defined panels, that is possible as well. So for panels, amplicon systems, right, there's sort of known ones, whether it was Fluidigm, AmpliSeq, and RainDance, and the power of these amplicons is you can be able to sort of use smaller quantities, but the problem is it's a little harder to sort of look at copy number changes, but that's sort of less of a concern within sort of a genetics as opposed to cancer. For exome, there's whether the capture that's used for exome is mostly we use Nivelgen and Trusic and Agilent. Well, most of our sites actually sort of use the Agilent-based technologies. But then as more and more discoveries are made, it is possible that then panels can be developed that's able to not only sort of having to do the whole thing, and eventually the clinical testing can be done for specific smaller types of sort of capture sets. And these are some examples, whether Emory or Baylor, developing smaller capture sets. But that's really, in terms of early stages of these genetic testing, you don't know what to capture. So starting with exome was probably the best way to do it. But as you know better, and you have a phenotype where you have suspected genes, then capturing with a panel would be a way to sort of to go there. In terms of the technology, I think most people on these calls would understand that there's been a tremendous growth in terms of sequencing, whether it's sort of first-gen sequencing of Sanger and then second-gen sequencing. All these companies, of course, sort of Complete has now been purchased, and Helicos is not as sort of viable. But the main ones, I think, like everybody does, is mostly based on sort of Illumina technologies, which is Lexo, and the Torn based on sort of live technologies. Most of our sites have been using more sort of Illumina-based technologies. But of course, we know that there is sort of onslaught of third-generation sequencing that's sort of coming on board, although most of them have not been really sort of commercialized yet. But we're sort of excited about these new technologies, especially for rare diseases, whether it's someone like a Nubio or Oxford Nanopore, and we really believe that as it becomes wider adopted and it becomes easier, a clinical testing that can be sort of distributed and done fast. So about sort of informatics, how are sort of the pipelines are used. In terms of informatics, the rough outline of informatics, right, starting from alignment to variant calling to filtering, annotation to clinical reporting. So some of the shown here is all the different sort of permutations, whether it's sort of done in the cloud or local. I think most of our sites is sort of, you see that sort of in the orange, sort of the locally is sort of done the alignment and variant calling, whether it's NovaCraft, NovaAlign, or BWA with alignment, and then SAM tools and GATK most likely, and GATK 1.6 for variant calling. So that's actually mostly sort of done locally and is able to reduce a lot of the file size and sort of a lot of the computational power if it's done locally. Although some of our partners have been able to do it in the cloud, but most of them have to sort of be able to sort of do that locally. Then the second step then is what we have seen is actually that stuff is local, but then the later with the filtering and annotation sometimes is done in the cloud. Well, some do it locally, but some uses sort of tools such as Ingenuity or Golden Helix to be able to do that. Many also do it locally and just sort of using simple scripts to be able to do that. So that's sort of one model where you split up between the variant calling stage and the filtering stage of how it's done. And then there are systems where they combine the two, which is shown in purple, and ultimately there are ones where it goes all the way to clinical reporting. The the challenge of clinical reporting is that you can't just sort of, make look at what's the output and specifically just say that. It needs to be databases that are known to be, verified in a clinical context. The data which you report is because they're going to be acted upon clinically, really has to be databases built on human data, and the levels of confidence, you really need to be able to specify a lot. So there's that extra step with clinical reporting, is a very, very critical step, although sort of shown as a small step here. This is one of the largest barriers right now. Because a lot of sites can do sequencing very simply and can do the alignment calling and, to figure out, depending on the genetic profile, what are the top genes. But to turn that into the clinical reporting is sort of the main hurdle. So how is sort of filtering done? And I think the pipelines are sort of very similar between even all our different sites. And this is sort of a general model between the different sites of how it's done. So whether it's usually what have happened is taking all the variants that you have, taking the known variants to sort of remove this so that you only have the rare variants. So whether this is using sort of more public databases or curated databases of known variants, such as Seattle SNP or whether Cosmic, or to be able to take those. Some people are able to sort of annotate with known SNP locations and be able to annotate that. Then this next step is going to be very important in terms of the genetic, and this is where a large of the filtering sort of occurs, and where it's important to have family members to be able to do that. And a lot of the filtering occurs in this step, going through genetics. Then once it's filtered, then the annotation and the database to be able to do that is going to be very, very critical of how you're able to do that. So whether people use HTMD or similar types of databases to be able to do that. But to be honest, there's still a great opportunity for someone to develop a really quality clinical annotation database for this, or whether something like NIH using ClinVar. Then there are different prediction algorithms that people use, whether it is something based such as ANOVAR or SIFT or polyphens to sort of predict, and sort of the jury's really out of how effective these are. But I think it's helpful, I think, for most of our sites to be able to sort of use this type of information as they're thinking about it. And then ultimately, there's sort of a manual step of curation and selection to be able to fit whether the phenotype is similar to, potentially similar to what's predicted by the genetic sequence. So this is sort of a general filtering strategy. Different sites have sort of slight modifications of this, but in general, this is a pretty good strategy to be able to do that in terms of. Then there's a big question of sort of how then it's sort of reported out, and I draw this sort of on the two axes. Is on the x-axis where it's how open or closed the system is, and then on the y-axis is going from research to clinical. So on the very, very bottom, the quadrant where it's closed in research, that's where traditional research lies, where it's done in a research environment and you don't sort of report it back. They can go a little bit up. You see that there's sort of area of clinical research where some of that information is relayed. And then you see there's a sort of some arrow there drawing past the x and y axis. Is some sites then do the sequencing in a research context and then validate using Sanger, CLIA, and then be able to report. For more open systems, you have companies who like the Personal Genome Project, 23andMe, that are able to sort of provide it all the data. Of course, the most extreme is Personal Genome Project, that's able to provide all the data in an open and source manner, and proper consenting and all that the PGP does is able to do that. But on the other quadrant, where it's clinical and open, this is where ultimately where everybody's sort of looking towards. You have clinical labs at academic universities such as Baylor and Emory, or UCLA, or you have companies like GeneDx, Ambry, and Vitae, to be able to sort of report a smaller subset. And then only if you ask, you're able to get sort of a little larger data. But in terms of the sort of data, in terms of I wrote these are sort of two companies of recently as pilots now been acquired, but PGDX, where it's sort of they're able to provide sort of a lot more sort of information, and more companies now are providing this model where they're returning a lot more data than only potentially the gene. They potentially even give the entirety of the data. And of course, this slide is very, very controversial of what to report back and how to report back incidental findings and how much the patient should really sort of have. So the next question then, if it's sort of really done as clinical sequencing, how is reimbursements done, and is it possible there? And what are the costs? So this is just a sort of brief graph of the cost and reimbursement. And of course, so in the past, sequencing, have, such as sort of BRCA test could be $5,000 to $7,000 with Myriad, or NGS sequencing, and that's still between under the 10K. And then there's some panels that are over 10K. So even just for one gene, a series of genes for one disease, it could easily go past 10K. But then other diagnostics clinically, whether it's a CT scan or MRI colonoscopy, they sort of fall in that range as well. And then you sort of think about, you want to sort of compare it to a week in the IC, in the NICU for a child that's born in NICU, easily sort of over $10,000, or a year of orphan disease drugs. So when we're telling people who are reimbursing and paying for this, the amount that usually right now for true exome sequencing around sort of $7,000 to $10,000 in this context, actually sort of often is a cost savings. And if you're able to sort of minimize hospital stay, or minimize use of, be able to target the use of these drugs, this is actually a strong case to be made for payers to be able to sort of provide reimbursement there. So as we think about sort of all these technologies, this is actually a very, very important slide. It's not sort of simple and clear cut and just sort of input the sequence and out comes the answer. And I'm going to sort of walk through this slide sort of a little bit sort of in depth in terms of thinking about CADnets. And what are sort of the barriers for this to be implemented on a large scale in all the hospital centers for insurance to reimburse it, and for this to be a largely adopted test, and what are the things that's preventing that? Number one is there's really sufficient power of annotation. So sometimes there's not enough families, there's too few of the same disease, or when you find the gene, the annotation isn't there. So there really is sort of a lack of databases that's able to annotate every single position, every single gene in the human genome, and we still don't know a lot of what the genome does. So even if you find a large panel of mutations at different aspects, you really don't know whether your conjecture, and that's all it is after your filtering, is true or not. You need sort of multiple families, and it requires a lot more information for that to be used there. So that's just databases and whether that's sort of sufficient. Secondly, sort of be able to sufficiently cover the genome. So there's sort of a lot of knowledge about sort of the first exon with high GC content, and then how that it's very hard to cover, and companies like like Personalis have been able to start to solve those problems. But still, coverage is going to be an issue, and if your gene of interest and mutation of interest is not correctly covered, whether it's sort of missed by your capture protocol, or whether your informatics is unable to sort of call that position, you can sequence all you want and you'll be missing that. So that coverage is going to be important. Number three, I think right now the pipelines are very strong for single nucleotide mutations and single nucleotide changes, but not still really developing in terms of copy number alterations, translocations, and other types of genetic sort of changes there. So if your pipeline is good at just sort of capturing SNVs, you may potentially be missing that, and as well as I think sort of include there sort of indels. So we know many different algorithms to be able to try to be able to call indel, whether it's sort of large indel or small indel, and that's still an important thing to sort of be able to take in consideration as you're doing these sort of sequencing methods. Furthermore, if you just do exome, of course, you need to look at introns and RNA, all the stuff in intronic regions that you'll miss based on that. So how you sort of design the study and your hypotheses before that. Furthermore, even if you have the perfect coverage, even if you have the, it's in your gene of interest and it's a SNP, and there are potentially sort of UDP or there is multiple genes or mosaicism. So looking at sort of potentially sort of non-Mendelian genetics is, you need to consider that, or even non-genetics, whether it's an epigenetic factor or microbiome. So all this must be taken into consideration to be able to do that. So really good sort of filtering, even sort of a priori, to make sure that this is thought to be a genetic test, a genetic disease is going to be very, very important. That's why I sort of talk about those criteria to be made, whether in a group context or individual context. Lastly, I have actually many, many more, but these are some I list out, is even in the algorithms that are currently are still being developed, right? Whether alignment, what is sort of good alignment? As we have longer reads, you can have better alignment, but that's still, there are better programs to be able to do it today. Variant calling, I think there's a lot of, with SAM tools and GTK, but there's, other initiatives, at Broad, be able to do group variant calling, and new algorithms be able to do that. And currently they're still, missing a lot of sort of variants in different ways, especially when there's sort of sequencing errors, or sort of lower sort of sample sizes. So the tags that you find are sort of lower number in terms of coverage and how you're able to sort of call a variant in that context. So this is sort of a quick sort of view of what exome sequencing and how it's going to be effective or not. This is a hype cycle that's used in technology circles. It's called the Gartner hype cycle, where in the beginning there's sort of technology is invented, then there's a lot of peak of inflated expectations. And I think we're sort of at this moment, and we're actually maybe going through the trough of disillusionment, that we know that a lot of diseases are not going to be all figured out, but there's still lots of exome projects being done. But then I think as we are able to really sort of nuance and finesse and knowing what we're looking for, we're going to go up that slope of enlightenment in terms of and implement this to sort of the plateau of productivity to be able to do there. But the thing is, this is all just really the beginning of these methods of sort of sequencing. Sequencing can only sort of do a very limited amount, and these are some of the technologies that we in-house are starting to sort of partner with or doing. Be able to validate. And this validation is going to be incredibly important in terms of figuring out what these mutations mean and what these genes mean and to really bring value to the sequencing. So for example, I think the picture here is shown of a mouse model. So model organisms to be able to recapitulate the disease phenotype and to really understand like whether it's really that gene that's sort of causing that. We're working with companies like Genomics and be able to sort of do that. Secondly, there's going to be on top, there's a picture of a microarray, but using other modalities is going to be important. So whether people are using RNA-seq to see whether that mutation is actually expressed in the tissue of interest, or looking with proteomics, whether there's sort of functional changes in the protein, whether it's ability to metabolize or whether it's a phosphorylate, dephosphorylate, to be able using higher throughput functional genomics methods to be able to annotate and understand the effects of these mutations. This is going to be extremely, extremely important to be able to do that. In addition, I think the picture here I would sort of put with Adgene is there's going to be need for sort of drug screening. So whether is even if you know the function, what are some of the existing drugs potentially through repositioning, repurposing that could help? For the famous cases, for example, with the berries, it's sort of missing neurotransmitter, and that's able to sort of provide cure very effectively there. So drug repurposing is going to be a very, very sort of important aspect there without having, for rare diseases, to go through those sort of long pipelines with common diseases to be able to do that. The next point here is sort of red in terms of growing cell lines. So without having to sort of do the actual experimentation on the person, having the cell lines and then to be able to do it experimental that for individuals is going to be important. So with the development of technology such as the iPS stem cell technology, where you can test these sort of existing drugs in these cell lines or understand the function in these cell lines and to use that as a system to be able to do that. And lastly, I just to show a general picture, you see a sort of a scientist holding up a beaker, is that there is an entire sort of large amount of functional study that's going to be important, as well as sort of leveraging all these additional technologies, model organisms, to be able to understand. So we are really at the very tip of using sort of genomics for interpretation, especially with all these clinical and exome sequencing that's done by companies. It's still very much sort of very hard to be able to definitive to say, oh, this is what exactly is able to cause it. So as more and more of these sort of functional annotation is done, there is an ability then to add to those databases for further prediction and functional annotation, and that database of interpretation is going to be very, very important, and having that is critical. That infrastructure is critical, and that database is critical for exome sequencing to be really adopted on a wide scale, as opposed to sort of in just as a research context. So here at Genomics, and our sort of small nonprofit consortium, where we will continue to fight, really. The 25 million patients are out there, all looking for answers, all looking for diseases, and for us to be able to help them with diagnosis and help them with the subsequent annotation, and ultimately, I think these parents don't give up until they find cures, and we seek to come alongside them every step of the way. So there are a lot of people that have been sort of critical, and we have over 100 scientists working with RGI, including on the left here, this is some of our core members, and on the left are core members, and on the right a lot of our expert panels who are able to not only provide sequencing understanding, but also in terms of what after sequencing in terms of functional annotation. And then most importantly, we want to thank parents, patients, and their communities to be able to sort of do this and partner with us on this. So thank you. That was sort of a quick overview of what we do and the mechanism we do that. If you have interest in collaborating or slides or views or questions, that's my email. Feel free to contact me there. So if you want continued education credits, this is the website to be able to do that. And now I'm open for questions. So looking at some of the questions, one of the first questions is, how exact is the genome landscape of human breast cancer for understanding disease? If we understand it, why do we fail? Sorry. Let me read that once again. So this is from Noble University, who asks, how exactly is the genomic landscape of breast cancer for understanding disease? If we understand it, if we understand it, why we fail to act for precise diagnosis or treatment? If we don't understand it, what's the benefit of research? So the question is the dark matter of DNA and whether this sort of actually matters for breast cancer. So in terms of cancer, our initial sort of sequencing is actually very, very interesting, seeing that all cancers are going to be very, very different. And what's going to be important is to understand the context, be able to sort of understand the context of these sort of heterogeneity, and based on what the mutations they have, then be able to sort of assign the correct medications and therapies for that. And the big thing is there is no current sort of, a lot of the targeted therapies are still much sort of in development. So as more and more different cancers are sequencing, whether through the TCGA or International Consortium, all these different studies, we'll be able to better appreciate the heterogeneity person to person as well as in person, to then start to be able to sort of, to quote, the tailor-made for therapies for different sort of cancers. So the landscape is not uniform. It is not one and the same. It's going to be different for different people, and thus for us to be able to start characterizing this on a large scale so that we can understand the individual variations is going to be very, very important. There's a lot of very great work, especially in sort of breast cancer, to do that, but for all cancers, as more and more different tissue types are characterized and are characterized and are understood, we're able to then sort of treat sort of future patients. Another question. The second question here is in terms of filtering strategy, prediction and then creation. So if prediction is not 100%, what will be the status of the curation? That's a great point. So in terms of the strategy, the filtering strategy, there's sort of two methods of doing it. One is to do it sort of, you literally filter, and it's no longer in that next step. And some people do that. Whereas other people have sort of given a more sort of a Bayesian approach where it's sort of less likely, using different characteristics to be able to do that. So the prediction usually is, so using, mechanisms such as sort of SIFT and polyphen, where they're predicting, they're predicting specifically, how deleterious this is, and then it's actually going to be very different between different algorithms and existing software programs such, Anavar and other program tools are able to actually to use multiple prediction algorithms to be able to see. Other ones actually are able to sort of leverage protein structure to say, whether it's hydrophobic, hydrophilic, and be able to then make an assumption that, if it's more in the core of a protein, it's sort of more disruptive. So the prediction is not one of a filtering strategy, but then it's to sort of help understand, to be able to help understand, what of these potential sort of mutations are causing. So then when you get to the curation step, some people are able to just sort of take the ones that are predicted deleterious, where other people are also able to then look at all the different mutations and say, what, even though that this is predicted to not cause that much effect, but it's in a gene that's potentially very much important. So let's sort of dig in a little bit more in terms of looking at it. So those methods will be able to take into consideration of the different prediction algorithms to be able to sort of assess which of the many sort of mutations that exist are the ones sort of causing the phenotype of interest. Another question here asking about, are you interested in finding treatments for single families with rare gene disorder? Definitely. So Rare Genomics was set up specifically to help families that are unable to be helped by other mechanisms. So scientifically, if you think about our setup at Rare Genomics, it's probably one of the worst scientific setups to think about, right? Helping single families is very, very difficult. What you want to do is actually help large families, large cohorts, all with the same phenotype, and do large projects to be able to do that. And the fact is, yes, a lot of people are doing that, and it's great that they're doing that. And we definitely think that's sort of the best efficient use of sort of scientific funding there. However, what if you don't fit into that? You don't fit into these large categories, and that's when the single families is going to be important if they don't have support structures helping them. And that's why sort of Rare Genomics we were founded to really help the people who are not being helped, and the families that may not be sort of as attractive scientifically. Then we equip these families with the tools so they can push forth themselves first, and to not only to look for diagnoses, but also look for potential, the function of this, to create iPS stem cell lines, to create model organisms, with the hopes of that they will push this research far enough that it will get the interest of institutions that will be able to sort of carry this deeper, or if they're really lucky to be able to actually find something that is going to be actionable right away. So in terms of that question, we are definitely very interested in finding single families with rare gene disorders. And our big shift now is there are a lot of families that already have been done sequencing, but they just have a gene and they don't know what to do next. And maybe that gene is not even studied at all. And we're in the middle of creating systems and infrastructure, partnering with companies that does contract research organizations and crowdsourcing to large scientific groups to be able to sort of help these families one by one. So another question, I'll do this question firstly quickly. Someone's sort of asking whether there's a possibility for a postdoctoral opportunity. So right now we are a completely volunteer-run organization. So if you're sort of interested in sort of volunteering your time and working, so we have a large number of scientists in all different aspects of academia, from postdocs to PIs to graduate students, all contributing in different ways. So if you're interested, feel free to sort of email me, and we can sort of talk about what way you can contribute, large or small. So unfortunately, we do not have sort of postdoctoral opportunity. Eventually, we would hope to have that. But if you're sort of interested in volunteering, we're more than happy to do. Next question. Whole genome and sequence the whole genome and exome sequencing for good diagnosis must fight in many different scientific fronts. Which front line of this battle will be #1 priority for whole genome sequencing? Yes, this is sort of many different ways to be able to sort of attack this. And I think right now what's very important, I think that the algorithms to be able to detect and to call single nucleotide differences is pretty much sort of there yet. So methods to be able to do the other types of sequencing. So those are ways to be able to do this. So which is what's I think sort of most pressing here is to have good databases to be able to sort of annotate the different clinical mutations. So that's going to be very important. So I think if there is a possibility for large initiatives where we're able to collect large amounts of good exome data and have it be annotated with good phenotype and phenotype and sequence, and I think there's international efforts to be able to do that. Then at the end of the day, the interpretation is going to be based on the other sequences that are available. So for international efforts to be able to come together and build those databases, whether it's something similar to ClinVar or others or PGP, I think that's going to be the sort of most pressing to be able to have a central place for all that annotation to live, so that we know every single mutation, every single gene, what that sort of is, the effect is, and then how we're able to do that. A few more questions here. Question here: what is the neurogenic effects of different disease? Hold on one second. What is the neurogenic aspects of different disease in clinical research of whole genome and exome sequencing? Can you ignore the brain and neurons of connectome in different diseases? So yeah, I mean, genome sequencing is only the beginning, right? Of course, in brain disease, the connectome is going to be very important, and on a cellular level, that's going to be important to understand, not just in terms of genetics. So there is a lot of actually advancement in terms of exome sequencing, whether it's mental illness, schizophrenia, Parkinson's, in terms of exome sequencing. But this is one of the many aspects of total understanding of that disease. So yeah, so it's important to to look at other aspects. So that's why the Connectome project is going to be very, very important as well, but as a complement, a modality to be able to do that. This one, a few more questions. Someone asked in terms of the programs that used to detect hydrophobicity and I know whether it's hydrophilic. I think the program is called CHASM, CHASM. I think Rachel Karchin does a program that does that. But there's a lot of sort of structural prediction of effectiveness of structural changes, actually how mutations cause structural changes, and mapping on existing PDB structures and solve crystal structures to be able to do that. So I think the one that we've used is CHASM. So it's one of many, many different characteristics to be able to do that. Question. How many previous undiagnosed rare diseases have been assigned to a causal gene through RGI? Yeah, no, that's a great question. In terms of the difficulty is how certain. We've gone through lots of patients and of diagnoses that we return back. The question is how many of them are we completely sure that that's the gene? And for us to be completely sure, probably very, very small number. But there's ones where we're almost very certain. For example, with Robert, what happens is it's a stop mutation in one allele and a truncating mutation. It's truncating in one allele and one is sort of inherited from one is de novo and one is sort of a nonsense mutation already that has been inherited. So it's sort of the classic sort of two hit there, and it's in a gene that has known to be associated with a disease, so that's sort of easier to call. However, so Robert, there's a known disease. However, let's say Robert's gene is in another gene that's completely new, which is what the question asker is. And then, subsequent research is done. So for a few, actually probably a handful of our patients, we're pretty sure we sort of found something new. We're doing the subsequent experimental research to be able to sort of validate that. So yeah, I think that's sort of an evolving number, I guess, for that. Well, thank you for all your questions and sort of joining our conference here. And hopefully this is helpful as you sort of think about sequencing not only in a research context, but in a clinical context, and figuring out ways to be able to help these patients with rare diseases. Again, if you have any questions about anything, feel free to email me. And I do work in also cancer as well as rare diseases, so happy to be able to, and hopefully we'll be able to sort of work together to solve all these 7,000 different diseases that affect the millions and millions of patients out there, and able to help them one at a time. Thank you very much.